Welcome to the Immagina Biotech podcast, powered by artificial intelligence and created for educational purposes.
In the fast-moving world of molecular medicine, the challenge, as always, is finding those truly robust, novel biomarkers.
Right, and identifying drug targets that have so far been completely unreachable.
For a lot of researchers, it feels like searching for a microscopic needle in a vast genomic haystack. So our mission today is to give you the map and maybe the magnet.
Exactly. We're going to focus on a critical yet, a largely unexplored layer of the transcriptome.
Okay.
We're talking about the 3-foot phosphorylated RNA fragments, or 3-Ph.
And these aren't just junk RNA.
Not at all. They're highly specific, active hallmarks of critical biological processes. They operate inside the cell, but what's really fascinating for diagnostics is that they're also secreted to biological fluids.
Which opens up a whole new frontier for non-invasive discovery.
Precisely.
And we're going to explore the proprietary methodology designed to unlock all of this, the DOORS platform. It's engineered not just for biomarkers, but also to find entirely new classes of RNA-based therapeutics.
At its core, DOORS is all about integration. We had to combine a really validated molecular methodology with some very specialized, powerful data mining.
You need both.
You absolutely need both. This dual approach lets us precisely detect, identify, and crucially, interpret these elusive 3' RNA fragments, which, standard sequencing often just misses or throws away.
Okay, so let's unpack this a bit.
Yeah.
Why focus specifically on 3' RNA fragments? I mean, what is it about that specific chemical modification, that phosphate group at the 3' that makes it so much more interesting than, say, full-length transcripts or even microRNAs?
Well, what's fascinating is their origin. Unlike, a stable messenger RNA, these fragments aren't just products of standard transcription. They primarily come from dynamic RNA processing, specifically from the enzymatic cleavage of non-coding RNA.
So things like ribosomal RNA, snRNA?
Exactly, and critically transfer RNA or 2RNA.
Yeah.
Because they're the result of an enzyme cleaving something, they're like molecular exhaust.
Fingerprints.
They're fingerprints of active disease processes happening right now. That makes them far more dynamic indicators of pathology than just looking at simple gene expression levels.
That distinction, dynamic activity versus just stable expression, that feels really key. We already know the altered expression of these fragments is implicated in some pretty major diseases.
Oh, absolutely. Cancer, viral infections, neurodegeneration, the list goes on.
But capturing them must require some serious technical work. So how did we build the technology to isolate and quantify something so specific? What are the two pillars it stands on?
We knew we had to be precise in the wet lab and really intelligent in the dry lab. So the first pillar is DART RNA. That's the technology that enables the selective capture and precise quantification of these fragments. Then that feeds into the second pillar, PROC, which is our machine learning-driven tool for the sophisticated interpretation and data mining of all the hits from the sequencing.
You really can't have one without the other.
You can't. You need precision capture, and you need intelligent, targeted analysis to have any success in this space.
Okay, let's zoom in on DART RNA for a second. We talk a lot about the novel enzyme. What does that enzyme actually let us do that a standard sequencing kit just can't?
Well, standard sequencing protocols really struggle with 3' fragments. That 3' phosphate group, it physically inhibits the ligation step you need to attach the sequencing adapters.
So the key step just fails.
It fails. Our enzyme is a breakthrough because it overcomes that inhibition completely. It's part of a dedicated sample prep workflow using this novel, internally developed enzyme that performs 3' ligation with incredibly high efficiency and specificity.
And that's combined with specialized linkers.
Yes, and together, that ensures we're only capturing and sequencing the true nucleus-cleaved fragments. We're ignoring all the background noise.
So that specificity is what elevates this from just another RNA-seq experiment into a highly targeted fragment assay.
That's the perfect way to put it.
So the enzyme gives us that molecular specificity, but then the data analysis needs to be just as sophisticated. That's where PROC comes in. For someone listening who might not have a dedicated team of bioinformaticians, how does PROC make this easier?
We really designed PROC with the end user in mind. Its value is, I'd say, accessibility, speed, and comprehensiveness.
So it's not just for bioinformatics experts.
No, not at all. You can use it without extensive experience. It analyzes these massive data sets from DOORS. Sequencing runs very quickly, and it offers a full tailored feature set specifically for finding those subtle patterns within the 3' fragment data.
It's the engine that turns the raw sequence reads into something actionable.
Into actual biological insights, yes.
So if we connect this whole integrated platform DART RNA sequencing feeding into PROC analysis to the bigger picture, I mean, this sounds like it opens up some incredible scope for clinical and preclinical research.
It absolutely does. This platform allows us to precisely identify, quantify, and compare 3 phospho-RNA features across, well, pretty much any condition or tissue.
And that translates directly into identifying fundamentally new RNA biomarkers for all sorts of diseases.
Yes, and it also extends into therapeutic development. For targeted therapies, the precision of these markers means we can develop companion diagnostics.
To find the patients who will actually respond to a drug.
Exactly. You can reliably identify which patients are most likely to respond, which reduces wasted therapy and, of course, improves patient outcome.
And the dynamic nature you mentioned, that must be key for monitoring.
It's crucial for longitudinal monitoring. Once the therapy starts, you can monitor disease progression, you can predict recurrence, and track the patient's response to treatment over time.
And after all that high-throughput omics screening, there's a final validation step, right?
A critical one. We validate the strongest marker leads using a novel targeted qPCR method. This step makes sure that the omic findings are robust and scalable before they move into clinical translation.
So how adaptable is the whole platform?
That's where the usability really shines. It's designed to be fast, it offers flexible outputs, both the big OMA sequencing and the targeted qPCR endpoints.
And it detects multiple markers at once.
Yes, and it accepts all inputs, whether you have a precious solid tissue sample or a non-invasive liquid biopsy.
And it works with existing lab equipment.
Totally. It's completely hardware agnostic, so it fits into the readout platforms labs are already using globally.
Plus, we're not starting from scratch here. We already have a clinical database for several diseases, including melanoma, SMA, and importantly for today, cutaneous squamous cell carcinoma, or CSCC.
Right, which gives us existing validated lead candidates to work from.
So to really show the power of this, let's dive into that CSCC proof of concept study.
Let's do it. So CSCC is the second most common non-melanoma skin cancer. While it's usually treatable, its high recurrence rate means there's a constant, urgent need for better non-invasive diagnostics.
So our team started with initial profiling using DART RNA sequencing on, I think it was 11 primary CSCC cells and 10 healthy keratinocyte or HK controls. And the data, well, it immediately confirmed the platform's utility. We ran a principal component analysis, a PCA, which is basically a powerful way to see how different the cancer samples look from the healthy ones. And the PCA showed a clear, distinct separation between the two groups.
Which confirmed that global 3P RNA profiling is an effective way to distinguish these cell types.
Then the clustering analysis pointed us to the most interesting targets.
It did. It identified 2 distinct groups of 3P RNAs that separated the healthy and malignant samples. One group had some LNC RNAs, but the most important component was the presence of 3P TRF's transfer RNA fragments.
And the specific finding that made us all sit up was that these 3P TRFs were predominantly upregulated in the CSCC cell.
Right, compared to the healthy controls.
But we knew we had to confirm this in actual patients. So crucially, we applied the exact same analysis to patient sing biopsies, 20 CSCC lesions versus 20 healthy skin samples.
And the results were entirely consistent with the primary cell data. This was the big confirmation that 3P RNAs have huge potential as markers, not just in vitro, but more importantly, in vivo.
So then we took that critical step into liquid biopsies, because that's where the real clinical utility is.
We performed 3P RNA profiling on plasma samples from CSCC patients and healthy donors. And through this comparison, we found 4 common 3P-TRFS GluCTC, ALA-GC ASPI-GC, and GLI-GCC that showed consistent cleavage patterns and differential expression across everything: plasma, the cells, and the tissue biopsies.
And that cross-platform consistency is the gold standard for a non-invasive diagnostic.
It is. The 3P qPCR validation in plasma then confirmed the downregulation of 3 of those fragments, GluCTC, ALA-GC, and ASPI-GTC in the CSCC samples.
Which lined up perfectly with the cell and tissue data, really underscoring their potential as highly specific, non-invasive biomarkers.
Now, this, of course, raises a huge question for anyone focused on the basic science. What is driving these dramatic changes in TRF expression?
Right, what's the mechanism?
We found that these cleavage events, they weren't because of a change in the expression of the full-length tRNAs themselves. Instead, they were linked to a lack of a very specific chemical modification.
The M1A modification at position 58 on the tRNA molecule.
That's the one.
That structural insight must have given us our mechanistic hypothesis.
It did. We dove deeper and saw a clear downregulation of the enzyme that actually places that modification. The methyltransferase job is to place that M1A modification, and that's essential for stabilizing the tRNA molecule. If that modification is gone,
tRNA becomes unstable and just falls apart.
it promotes fragmentation. Exactly. This allowed us to propose a really compelling molecular model.
Okay, so walk us through it.
In CSCC, we propose that the downregulation of one specific fragment bind to the messenger RNA.
And that binding causes the transcript to be degraded.
Right. So less protein means less M1A modification at position 58, which ultimately increases the rate of tRNA instability and fragmentation in the cancer cells.
Understanding that precise mechanism, it doesn't just give us a diagnostic, it gives us a target. And that target leads us to the next, and I think truly revolutionary application, using the DOORS platform to explore RNA-based drugs that target ribosomes.
That's right. I mean, the therapeutic potential of RNA-based drugs is immense. They can interact with proteins, transcripts, genes, reaching binding sites that small molecules just can't get to.
But there's a huge gap in the field.
A major gap. There are currently no known RNA drugs specifically targeting ribosomes, the central machinery of protein synthesis.
Targeting the ribosome is sort of the holy grail of translation control, so our proprietary approach, which comes directly from the specificity we get with the DOORS platform, it's designed to fill that gap.
We can use structured single-stranded RNAs to literally engage the ribosome body and modulate the translation process itself.
And what does that let you do therapeutically?
This opens up entirely new therapeutic pathways. We can design drugs that either stimulate translation, which has massive implications for things like vaccine adjuvants or rare diseases.
Where you need to boost a specific protein.
Yes. Or we can design drugs that inhibit protein synthesis selectively in certain cell types, which is obviously crucial for oncology and other highly proliferative conditions.
And this potential is already driving our internal drug pipeline.
It is. We're co-developing RNA-based medicines in vaccines, immunomodulation, and cancer. That includes both 3' ribosome modulator suppressor drugs to inhibit protein synthesis and activator drugs to stimulate it.
The same technology you use to find the biomarker is the exact same pathway used to design the therapeutic.
Exactly. So if we put this all together for you, we validated the ability to use 3' RNA fragments to find disease markers, to investigate companion diagnostics, and critically, to explore a brand new type of single-stranded RNA drug action directly on the ribosome.
So what does this all mean for you as a researcher or a clinician? You now have access to a fully integrated patented platform. It combines the power of that novel enzyme, DART-RNA-seq, the proprietary PROC AI pipeline, and targeted qPCR, all to explore 3' RNA fragments as highly dynamic diagnostics, prognostics, and pharmacodynamic biomarkers.
And this proprietary technology, it's protected by 3 issued patents covering the methods and the lead sequences. We've already identified validated lead candidates ready for the next step.
So we're offering the scientific community the ability to move beyond the established RNA classes to identify novel disease markers and drug leads.
To create precise companion diagnostics for targeted treatments, and to explore the mechanisms of entirely new types of ssRNA drug action on the ribosome body.
So here's a final thought. Considering the high stability of these 3p-TRFs in biofluids like plasma and their remarkable non-invasive detectability, how might this stability and specificity fundamentally change our current paradigm for monitoring treatment efficacy and predicting disease recurrence in the very near future? If we can actually track the molecular exhaust of active disease in real time, it shifts the entire curve of patient management.